Welcome to this explainer. You know, science is usually built on really solid foundations, fundamental rules that basically dictate how life operates at the microscopic level. If you've ever taken a biology class, you were taught these rules. They tell us how genetic information flows, how cells replicate, and just how life keeps ticking. But the thing about nature, it has this fascinating, almost rebellious tendency to completely ignore the very textbooks we write about it. And today, we're exploring a groundbreaking discovery that might just rewrite everything we thought we knew about molecular biology. So what happens when the most fundamental rule in biology suddenly has an exception? We're going to look closely at how rigid biology textbooks are actively being challenged right now by a tiny, mysterious bacterial machine. This little machine pulls off an incredible, almost impossible magic trick. It creates highly specific, complex DNA out of seemingly thin air. And in science, when something appears to just pop out of nowhere, it almost always means there's a hidden mechanism, some secret process we just hadn't noticed yet. But to really appreciate how mind-bending this trick is, we first need to establish the golden rule that this machine is so casually breaking. Okay, to understand the surprise here, we have to talk about the central dogma of molecular biology. You can think of the central dogma kind of like a highly secure photocopier, or maybe a precision 3D printer. The fundamental rule goes like this. Genetic information only flows in one specific direction. If you want to make a copy of DNA or build a brand new strand, you absolutely 100% must have a blueprint, and that blueprint is always a nucleic acid, either an existing piece of DNA or a strand of RNA. It serves as the physical template. Without that template telling the molecular machinery exactly which building blocks to lay down next, synthesis just doesn't happen. Life, as we know it, simply can't replicate. Or at least, that's what the dogma has told us for decades. So where did this rule-breaking machine even come from? Well, this discovery stems from an eternal, invisible war that's happening all around us every single second. It's the fierce evolutionary arms race between bacteria and the viruses that hunt them, which are known as phages. To give you an idea of the stakes here, phages vastly outnumber bacteria in the environment. It is a relentless, non-stop onslaught. And because these phages are incredibly lethal, bacteria have been pushed into a corner. Over millions of years, they've been forced to evolve bizarre, highly complex, and intensely creative antiviral defense systems just to survive. And the specific system we're looking at today, it's a defense mechanism unlike absolutely anything we've ever seen before. All right, let's dive right into the machine itself. Scientists call this complex DRT3. It's a bacterial defense mechanism that somehow pumps out long, repeating strands of double-stranded DNA to stop viral infections from taking over. Specifically, it manufactures a really unique sequence. It makes an alternating pattern of G and T on one strand and A and C on the other. Think of it like a massive chain made of just repeating letters from the DNA alphabet, over and over, GT, GT on one side, and AC, AC on the other. But the big mystery that had researchers completely scratching their heads was, how in the world is the bacteria perfectly manufacturing this highly specific alternating sequence without using a standard blueprint? We can actually break this biological machine down into two main engines working together. Now, these engines are known as reverse transcriptases. Normally, a reverse transcriptase is an enzyme whose whole job is to read an RNA blueprint and write a new piece of DNA based on that exact template. The first half of this machine is called DRT3A, and DRT3A follows the rules. It uses a non-coding RNA blueprint to precisely print out its half of the DNA sequence. But the second half of the machine, DRT3B, is a total rogue agent. It manufactures its DNA strand without any nucleic acid blueprint whatsoever. It literally completely ignores the central dogma. To figure out exactly what these two engines were up to, scientists used an incredibly powerful imaging tool called cryo-electron microscopy, or Cryo-EM for short. This technology lets researchers essentially flash freeze molecules and take phenomenally detailed pictures of them, right down at the atomic level. And what they found was a stunning hexameric assembly. It's basically a massive six-sided ring made up of 6 DRT3A proteins, 6 DRT3B proteins, and 6 strands of non-coding RNA, all perfectly locked together to coordinate their work. It's just a beautifully complex molecular factory, with each part playing a very, very specific role. If we look closely at DRT3A first, the imaging reveals that it behaves exactly like a conventional polymerase. It follows the standard rules of biology through classic Watson-Crick base pairing. That really just means it faithfully reads its non-coding RNA blueprint, specifically a repeating AC-AC-AC motif, and uses that physical guide to seamlessly match and print out a complementary GT-GT-GT sequence of DNA. It reads an A, it places a T. It reads a C, it places a G. It is a textbook, perfect example of biology following the established rules to a T. But here is where the mystery gets crazy. If DRT3A uses RNA to know what sequence to build, we have to ask a massive, paradigm-shifting question. How does the other half of the machine, DRT3B, know how to perfectly build its alternating AC half of the DNA? There is absolutely no RNA or DNA template anywhere near its active site. None. It appears to be working completely in the dark without any instructions. Well, the crucial answer is revealed right here. DRT3B uses its own internal structure as the blueprint. Instead of reading a separate RNA strand, DRT3B uses a specific sequence of its own amino acids, essentially its own protein body, acting like a template to force the incoming DNA building blocks into a perfect, alternating sequence. It physically kinks the DNA backbone so that the bases interact directly with the protein. It's exactly like how the physical, hard-coded shape of a 3D printer nozzle completely dictates the form of the plastic coming out. The protein is incredibly acting as both the builder and the blueprint at the exact same time. And this right here brilliantly illustrates the paradigm shift we're looking at. Normally, standard DNA synthesis requires a DNA or RNA template to dictate the sequence through classic Watson-Crick base pairing, where nucleic acids match exclusively with other nucleic acids to form the rungs of the DNA ladder. But with the DRT3 system, we are witnessing a newly discovered protein-templated DNA synthesis. It's actually using hydrogen bonds directly from the protein's own amino acids to select which DNA letter comes next. It just skips the middleman entirely. Let's put it all together and break down how this complex system operates step by step to form an actual deployed defense weapon. First, the DSAT3A engine operates normally, using its RNA guide to manufacture the long GT strand. Simultaneously, right next to it, the DSAT3B engine uses its own folded protein structure to manufacture the perfectly matching AC strand. Finally, these two separate, newly synthesized single strands find each other and anneal. They zip together perfectly, twisting into the final double-stranded repetitive DNA weapon that acts as the ultimate shield, ultimately stopping the invading phage virus right in its tracks. To really appreciate the molecular precision at play here, scientists pinpointed the exact amino acids doing the heavy lifting inside DRT3B. Specifically, it's the exact chemical shape of amino acids like glutamate 26 and arginine 253 that selectively grab the correct DNA building blocks. Glutamate 26 literally mimics the shape and function of the missing RNA blueprint. By forming specific hydrogen bonds, it creates a physical mold that perfectly fits an A block, while actively rejecting any other options. It's just an elegant physical trick that literally replaces the need for a nucleic acid template. Now, keeping a factory like this running takes a ton of energy, so bacteria don't just leave it on all the time. Much like our own immune systems that only activate when they detect a specific pathogen, this incredible protein-guided defense system is highly regulated. It stays completely dormant in the bacterial cell, quietly waiting. It only activates when a very specific attacking phage protein named ST61 enters the cell. The mere presence of this viral ST61 protein acts as the trigger, telling the DRT3 defense system to wake up, deploy, and start furiously manufacturing those protective DNA strands. You can see why this discovery completely blew scientists away. We are seeing information fundamentally flow from the physical structure of a protein back into a specific DNA sequence, a concept that wildly expands our understanding of biological information transfer. Not only does this directly challenge textbook molecular biology and the central dogma, but it also matters deeply for our future. Historically, whenever we find a radical new way that nature synthesizes or manipulates DNA, it almost always leads to massive leaps in biotechnology. This bizarre bacterial defense could eventually inspire wildly new tools for medicine, advanced gene editing, and the next generation of synthetic biology. And that leaves us with a really exciting and maybe slightly daunting question. If common bacteria have been hiding an incredible, rule-breaking protein-templated DNA synthesizer this whole time, quietly using it for millions of years to fight off viruses, we have to wonder, what other incredible mechanisms are waiting to be discovered inside the cells all around us? What other textbooks are going to need a complete rewrite in the next few years? Thank you for exploring this fascinating scientific breakthrough with me.